Okay, good morning, everyone. It's a pleasure for me to be here, and hopefully we can have a discussion today after the talk, and of course during the question session. So today, yes, I will try to give you a glimpse into the tree of life, how we attempt to reconstruct it, and how we attempt to make sense of it. So first of all, I wanted to just give you some background on my curriculum. So what I do, I study the evolution of bacteria and archaea using bioinformatics. So I'm a computational biologist. I study the mechanisms of molecular evolution and to actually try to understand the origins of biological functions. For instance, I've been working on the diversification of cellular machineries, those involved in virulence, for instance, and in motility. And nowadays I'm interested in the evolution of biochemical pathway and bioenergetics, and in particular, for instance, how lineages adapted to increasing level of oxygen after the Great Oxidation event. So to do that, actually, I study genome sequences, and to this end, I also do some software development and develop machine learning approaches to perform genome annotation, trying to make sense of the genome sequences that we obtain. So we'll talk about this later on as well. Yes, so actually I did my PhD in Lyon, and then I was a postdoc at the Pasteur Institute for 4 years. I did a short postdoc at the University of Vienna in Austria, and now I'm a CNRS researcher in Grenoble. Yes, so the outline of this talk. We will start with the diverse representations of the diversity of life, and then we'll talk about what actually brought molecular phylogeny. And then when these molecules actually became more accessible, what brought actually genomic information to our understanding of the tree of life and life history. Okay, so first of all, maybe I like actually the diverse representation. There is diversity in the way life diversity was represented itself. And I give you this actually one introductory slide. I won't go into the details, but if you're interested, there is a very nice review by Mark Ragan from 2009 about how diversity was actually represented, life diversity was represented a long time. And it goes, for instance, as early as the 14th century here, this representation, which is actually called the scale of nature, Scala Naturae, by Lull. And two maybe more famous representations that you have seen of actually categories of diverse animals by Lamarck. You have this more famous tree by Haeckel from the 19th century. And here actually what is interesting is that there is actually a difference between categorizing, classifying organisms, and actually trying to make sense of what are their evolutionary connections. So there is a difference. And actually this tree, which represents a tree of diverse willows, is actually trying to make sense of the evolutionary history and the evolutionary connections between those willows. And you see sometimes also representing life diversity can go through these network representations here, and here it's actually, for instance, dog breeds that are being represented here. So yes, we're talking about the tree of life, and actually one famous tree is this tree by Charles Darwin that was in the notebook on the notebook B. Actually in this tree what I like is actually that it represents two different notions in particular compared to other types of trees. It's the notion of evolutionary distance, where actually there is a sense of evolutionary distance, what is called in the note associated to this drawing, gap of relation, for instance, where it say that B and A in the tree have a gap of relation, meaning that they are somehow distant. And also with this actually branches here there is a sense also of the notion of extinct lineages, meaning that what we observe, contemporary organisms actually do represent only a portion of life diversity that has it evolved on Earth. And some lineages went extinct. So why a tree? Actually, it seems to be like natural representation for vertical descent. And most of life, and I hope my talk will actually convince you that most of life is represented by microbial organisms. And many microbial cells actually do replicate via asexual binary fission, which is actually more or less clonal reproduction. And it is illustrated here, the process of binary fission in prokaryotes, meaning bacteria and archaea is represented in more detail with also how it affects the replication of genome and the splitting of two cells from a same mother cell. And actually I illustrated in a very simple manner here with this one mother cell that actually will give rise to two cells and so on across generation. And actually, this is vertical descent, and we have this natural, actually underlying tree that emerges from these properties, where the genetic material is actually transmitted from one generation to another. And as you know, there are mutations that will accumulate. Some's going to be selected, some of us won't. Some lineages will go extinct, some of us won't, but in the end you can also have the notion of speciation that will actually arise a long time. So this is in blue what we could call a species tree. Okay, so maybe very briefly, because probably you all know that, or I don't know actually if you do know that, but how to read a tree. Actually, this is a tree that is rooted here, meaning that we know what is the most ancient branch in this tree. So this would represent here, these branches here would represent ancestral organisms, whenever the leaves here, the tips of the tree, would represent contemporary organisms. So as you go up in this tree, you go back in time. Also, I wanted to make this quick note about actually the notion of living fossils or archaic organisms, as we sometimes hear in news outlets. I wanted to emphasize that all contemporary organisms, they derive from a single common ancestor. Some may have evolved some features that diverge from that of their ancestors. Some others might have retained more features that stemmed from the common ancestor, but actually all evolved for the same amount of time because they are all contemporary organisms. So yeah, I just wanted to make this quick note, because we tend to hear about these notions and in the media sometimes. So how do we actually obtain a tree, and what is the underlying concept? And this was coined, the term of phylogenetic systematics was coined in 1965 by Willy Henning. And actually it relies on a simple notion. It is the fact to study shared and derived characters to build the relationships between organisms. It means that you will hunt for characters that actually would be shared by organisms to the exception of the others. So they have something in common that the others don't. So hence they might have a common ancestor to the exception of the others. So that's the basics of phylogenetic systematics. And yeah, so the fact that if a trait is shared by two organisms, it may have been inherited from a specific shared common ancestor. So this aims at explicitly reconstruct evolutionary relationships, and at first it was actually done using observable characters. So you'll see that everything is in the choice of the phylogenetic markers in the characters that you use to do this phylogenetic study. So at first, using morphological characters, and here is a small example. So let's imagine that you have observed 3 different types of characters. Here you have, for instance, the presence of vertebrates, of 2 pairs of limbs, of an amino sac. Okay, so those are the observable characters. You have data on this. And you have different candidate species trees that you want to actually evaluate. And you will use the distribution of these characters to evaluate one tree versus the others, to compare them. So you have candidate species tree number 1, where you have these 4 species. Actually, the shark and the bat share a common ancestor, with the exception of the bird and insect. So that's species tree number 1, first candidate. And species tree number 2, where you have bird and bat together, and then the shark that would branch, and then the insect. So then, what can be done? Of course, you start to map the presence or absence of the characters on the trees. So it goes the same, I mean, for the two, except that then, so the black dots represent the presence of the trait, and then you will actually infer the number of events in terms of acquisitions of the trait to explain the distribution in the tree. So for instance, here you have these 3 species harboring the vertebrates, and as they are all grouped together, it means that you have to involve only one acquisition of vertebrates to make sense of this distribution along this one tree. Okay, so this is one step for vertebrates. Then, if you do that in the other candidate tree, you see that the shape of the tree makes you infer 2 different events of acquisition of vertebrates to make sense of this distribution. So you can do that for all of the different characters here, and in the end, you can score actually the trees using the number of steps that it's required to make sense of the distribution of the different considered morphological characters in this case. And so you see here that, for instance, score for tree number 1 would be 6 steps, and then score for tree number 2 will be 3 steps. So in the end, actually, you can pick the most parsimonious tree, actually, the one that will involve less evolutionary steps, and it would be preferred as the one that would represent better the relationships between the species. So this is how we can actually use this morphological character. But if you want to build a tree of life, you may want to put all the organisms you know into it. So how to compare a sponge, for instance, to a human? So how do you do that, this morphological comparison? And even further, comparing a bacterium, Yersinia pestis, for instance, to a human, and goes on. And even at the level of prokaryotes, here you have these different cells, actually. Would you be able to tell me which are archaea or bacteria? They stem from 2 domains of life. Well, actually, you can see that some archaea are cocoid-shaped, just like some bacteria, and some are rod-shaped, but some bacteria are, but also some archaea. So it's difficult, actually, to find the right characters to compare organisms to perform phylogenetic analysis. And of course, goes the same how to place all of these different forms of life into a single tree, on the same tree. So this is where actually came into place the advent of molecular phylogeny. So the thing is that we have to find commonalities between organisms to be able to place them into a single tree. You have to find comparable traits. And for instance, here I'm showing you a typical bacterial cell and a eukaryotic cell. You have different sizes you've seen, and also different organizations. For instance, in the bacterial cell, the DNA is more or less free in the cell. Whenever it's packaged into the nucleus for eukaryotic cells. But there are these differences. There are also some similarities. And for instance, the genetic information is stored into DNA for both. And also there are some processes that are universal, like the ribosome, that are involved in the translation of actually RNA stemming from DNA. And those ribosomes are actually involved in the production of proteins from the genetic information. So these actually are common to the bacteria, and to eukaryotes, and also to archaea. So using actually some of the traits, it is possible to extend systematics to all different life forms. And actually, this is what was proposed in 1965 by Zucker-Kandel and Pauling, where they proposed that molecules could be used as documents of evolutionary history. And in this seminal paper, they actually proposed 3 what they called semantids by then, as they are proposed to hold genetic information. That would be the genic sequence of DNA, messenger RNAs, and proteic sequences. And you see that we can use this actually information to compare different organisms. And what is great about actually those characters is that they are very abundant as well. So we also need a lot of information. So those are abundant markers, at least in life they are abundant. Then the way we access them, they used to have, it needed some technological advances so that they would become viable. But also they are directly involved in the transmission, vertical transmission of genetic information. So it makes them very highly suitable to reconstruct the history of life. In particular, in 1987, this seminal paper by Carl Woese and colleagues actually proposed to use ribosomal RNA genes from the small subunits of the ribosome. So the ribosome is the machinery, as I said, involved in translation, the production of proteins inside cells. And these are ribosomal RNAs. Here you have examples across the 3 domains of life: bacteria, archaea, and eukaryotes. Well, they have a structural role, and they are actually highly important for the functioning of our ribosomes. So they are found in the 3 domains of life. And they were actually called the ultimate molecular chronometers in this paper. So then we have these molecules at hand that can be used to perform phylogeny. And actually this is where comes into place molecular phylogenetics. So the idea is to build a gene tree from sequences of different organisms, using bioinformatics, actually computational biology, and programs that are actually dedicated to that. So here is an example where the 16S rRNA genes are used on different species of bacteria. So first they have to be aligned, actually, meaning that the different positions will be put together, put in front of the other, so that they would actually match and correspond to ancestral position. So in the ancestor, probably here you have a T, you don't see it well, but you have a T everywhere in extant organisms, in contemporary organisms, so probably the T was also the actual ancestral base. So you make them coincide. Sometimes you may need to add gaps and so on, so it's not so easy actually to perform, because there are some modifications that can arise along time in these molecules. And then, once you have performed your multiple sequence alignment, can be based on genes or protein sequences, you use these programs for tree reconstruction, where there is a model of sequence evolution, and in general, the most, actually the state-of-the-art methods would be probabilistic methods that actually build proposed trees that would be the most likely given this multiple sequence alignment, so that you can make sense actually of all the different positions along the tree. And you obtain this phylogenetic tree that actually represent the relationships between the sequences in this case. Okay, so this was used to build this tree domains tree of life. And that was actually a first where it was possible to put all organisms, all extant diversity into a single tree, and that was performed in 1987, again from the 16S rRNA by Woese and colleague. And here you can see that you have 3 parts of the tree that came into place. And actually, what is interesting here is that you see that this was the formal denomination of archaea. They were thought, probably based on morphological characters, to be part of bacteria, but this tree actually placed them in a separate branch. So this is what we would call a 3-domains tree of life with bacteria, eukaryotes, and archaea. And in 1990, yes, this one tree actually came also with more sequences available and more diversity covered, and this is where also was the first formal description of the 3rd domain of life of archaea. You see now it's been renamed from archaebacteria to archaea. So yes, there are 3 domains of life, and now you can start planting all these different organisms onto it and try to make sense of this diversity and evolutionary connections. So the 3 domains view of the tree of life was a revolution in life sciences in our understanding of life diversity. And also more and more gene sequences became available with the advent of sequencing techniques. And what people noticed is that there were gene phylogenies that they would obtain. There were as many gene phylogenies as gene markers. So would that make sense? Why would these gene phylogenies differ? So people would tend to think that they should reflect species tree, and there is one species tree, so why would these gene trees be different? Actually, there are several reasons for that, and the first one is for methodological reasons. I won't enter into details, but it is actually difficult to model sequence evolution. It is difficult to find enough signal to reconstruct sometimes relationships that are as old as 2 billion years old. And also there are biological reasons for that. So let's take this as an example. This is a species tree with different types of bacteria that are here drawn. And now let's see, depending on the gene history, how will turn out the gene tree. So if the gene is transmitted vertically from generations and generations, from the ancestor to the contemporary organisms, well, you would assume that the obtained gene tree here would be identical, will have a similar topology when compared to the species tree. So you compare the gene tree and the species tree, and you can make some assumptions on the process. This gene was transmitted vertically, it reflects the species tree. But actually, the genomes are dynamic, and genes actually have different histories. And I give you here the example of the bacterial genome. So this is represented as a simple circular chromosome. I'm showing only a few genes, but actually there could be around from 1,000 to 3,000 on average genes in a bacterial genome, whenever you find around 20,000 in humans. So I just drawn as these boxes the different genes along this circular chromosome. And actually the bacterial chromosome is dynamics, and there are some events that can happen along evolution. For instance, genes can be lost. And there are different processes for that. You have what is called gene excision, where you will lose a part of the genetic material, and you have also what is called pseudogenization, where the gene would actually disappear as it will accumulate mutations, and it won't code for a protein anymore, and it would be pseudogenized. And there are also other processes that account for gene gains. And in this case, there are several types of ways to acquire new genetic material. One is actually called gene duplication, where a gene copy is duplicated, and it turns out that you have in the end 2 gene copies, which is a substrate for molecular evolution. You can have evolution of a new function for the gene based on this substrate of this new copy of the gene. And you can also acquire new genetic material through lateral gene transfer, and it can be from another organism, for instance. And for instance, you have this famous example of antibioresistance that is transmitted between bacteria via this process of lateral gene transfer. So basically, for instance, you will have this resistant bacterium here with this pink gene that would be involved in resistance to antibiotics. Through lateral gene transfer, and there are actually different mechanisms that actually make this possible: conjugation, transduction, transformation. Well, you can have the transfer of this one gene to another gene that was at first a sensitive bacterium, and that became actually resistant to antibiotics as well because it acquired the function through this lateral gene transfer. And actually what happens is that when you build a gene history that includes lateral gene transfers, you can actually make a diagnostic of this event based on the gene tree shape. So I explain here in this case. For instance, imagine that you have a transfer from this one species to this one. So this is the species tree, as a reminder. So you have vertical transmission for the gene on all different branches. Then you will end up with this gene tree, where actually you will find an abnormal tree position for the receiving species. So the gene was donated from this species to this species, and hence in the gene tree you will have the receiving lineage that will actually branch next to the most closely related sequence, which is the one from the donor lineage. You see, so you end up with a different gene topology. And in this case, actually, this abnormal tree position, you can study it systematically. And for instance, I have been developing such programs that will compare the shapes of the species tree and gene trees so that you make inference of evolutionary events. You can do the same also with duplications, with losses of genes, and you can try to make sense of all the genes history when you compare them and reconcile them with a species tree. So when people actually started to see that many genes' history would differ from that of the species, people would start to think, okay, well, is there really an underlying tree of life? Or is it this more like a little bit more messy situation where there were so many exchanges between different organisms and domains of life that it is even impossible to have an underlying tree structure? And actually that was a debate for some time. But also what we can see is that we don't have to make the confusion between the trees of organisms, the species trees, and the trees of genomes, and the gene tree. So this is actually summarized in this paper from 2009, where actually is shown a species tree. And now we have access to many gene sequences through genome sequencing. We'll talk about that later. And you can obtain what we call a genome tree by combining the information of many genes together. And of course, you will see that there are some branches that are actually abnormal and that will link and connect distantly evolved organisms. But still, you will have a hold of the underlying major signal, which is vertical transmission. So that would be how you can infer, actually, from genomes, you can infer species tree. And of course, the gene trees, as we said, they can differ from the species tree, but even sometimes it's very actually useful to make sense of the evolution of gene functions. So then, with all that, actually, as I said, there were technical advances that made more and more sequences viable. And actually, it was the beginning of the era of phylogenomics, where actually it's not single gene trees that are being used. And as you see, as you've seen here, actually, trying to reconstruct a species tree only based on the information of one gene marker doesn't appear relevant anymore. You want to combine as much information as you can get a hold with, and this is actually what happened with all genome projects that provided a huge number of marker genes for phylogeny. So people started to build these phylogenomic trees by combining the information of multiple genes. And actually, there were some methodological advances that would allow such analysis because it can be computationally heavy. And some progresses were made in the understanding of the diversity. But at that time, actually, most of the genome sequences would be from cultured organisms. So it would not really account for the life diversity. So this is something that was actually somehow called the hidden biodiversity, or the microbial dark matter, even though I'm not sure I would concur to these terms, but actually it is a fact that most organisms are not culturable, and it was then difficult to access to their genomic material and to sequence them. So how would we be able to evaluate the extent of the diversity on Earth, its ecological role, and how to decipher relationships between organisms if we don't have access to the genomes? So this is where metagenomics, environmental metagenomics, came into place. So usually, you probably have heard about it. Okay, so you go to a nice place. I used to have colleagues going to very beautiful place to sample. And actually, my first sampling trip was more into this pond, which was as interesting because we found very interesting organisms there. And it was easy to go there because it was just 2 metro stations next to the lab. So it was easy, accessible material. Anyway, so you go to sample environmental, well, here, for instance, you would take the water or sediments. Then you'd go back to the lab. Some people would actually try to perform enriched cultures. They will try to grow the organisms coming from the environment. It's not something you have to do. I think it's very interesting to attempt to do it, but it's a lot of work actually, and not always rewarding, but when it works, it's amazing. You can go also next just to DNA extraction. So you would process the samples, extract DNA, then you would go to sequencer, and you obtain what we call genomic reads. And from these reads, actually, you can process and go to this step where you can try to assemble the reads into a genome. And it's actually basically a DNA puzzle that you're trying to assemble. I won't go into the detail, but just because I'm a computational biologist, I want also to give you an idea of the techniques that we use. But basically, there are 2 big techniques to assemble genomes. One that is maybe more intuitive, which is called string overlap approaches, where actually you will try to connect the reads by overlapping strings. And then you obtain a bigger genomic sequence out of the small reads coming from the sequencing machine. And this is what we call a contig. And another approach actually is more efficient computationally to deal with huge data sets, which is called the Bruin graph approaches. There are several software that actually do implement both types of approaches. And actually, sometimes you just try to take the ones that will give you the best results with different metrics. Anyway, this is how you would assemble contigs. And then you see that, of course, you're taking environmental samples. It is like a soup of very many different genetic materials coming from different organisms. How to make sense of this? Well, to do that, you try to actually assess and reassign these different contigs you have built to a particular organism. And this is what is called actually genome binning. And here, as I'm showing it on this graph because there are different techniques to do that. I won't enter the detail again, but basically the idea of genome binning is to try to use, to find a common theme that would be actually a feature shared by the contigs of interest because they come from a single organism. For instance, they would tend to have the same content in GC bases, the same tetranucleotide frequencies. This would be like common features that might stem from a single organism. And actually, there are also some other approaches. And again, I won't go into detail, but for instance, if you have organisms that are found in 2 different samples, you may want to try to find the contigs which abundance will co-vary between the samples. That would mean they come from the same organism. So that's the basic principle for differential coverage binning. Anyway, I just wanted to tell you that all of this, actually, there are heavy methodological developments also being done so that we can make sense of all of this information and before we can have access to these genomes. So, so people actually try to sequence as many actually samples and sometimes even already viable cultures that they had it at hand because sequences costs lowered and computational powers were actually increased and the methodologies were viable to perform these genome assemblies. And for instance, this one initiative, that was called actually the Genomic Encyclopedia of Bacteria and Archaea, or GEBA, and this is a paper from 2017, sorry. people actually went to this strains collections. People have been actually collecting different strains and of like an archive of cultured organisms, and they have deposited them in strains collection. And actually, from this strains collection, many were not even, these organisms were not sequenced yet. So instead of going to environmental samples, these people actually thought that, okay, maybe let's start to sequence those organisms that we already know how to cultivate. I mean, to me, it makes a lot of sense. And you don't see it well here. The colors don't show up so nicely. But anyway, what you can see is that this is a tree of life, actually. Oh, this is, sorry, I think it's only bacteria. No, there are archaea. Sorry, archaea are represented here, and those are actually bacteria lineages. And at the tip, you can see that there are pie charts, and basically they would represent how much of this project, how much of the diversity was covered by this project prior to previous information in public databases of genomes. So this is in blue prior to GBA. You don't see it well, but trust me. So you will see a lot of red as well. So those would be the new genera added by the GBA project, for which genera for which the genome sequence will become available thanks to this project. So this is what they showed here also, where actually it is a way to represent the diversity and the number of cover strains. So they're showing the different type strains available, and what they say is that, okay, before our project, well, there was this amount of diversity covered, and actually they covered this amount of diversity after the project. So, sequencing of type strains, even though they were long culture organisms, it was nice to have finally access to their genome, and actually it helps to really accurately try to classify and to position them in the tree of life, these cultured organisms. That's the first thing. And also, they showed in the article that they could discover new biosynthetic gene clusters, which are actually have the potential to produce new metabolites, maybe even antimicrobial compounds, etc. So also these genomes hold interesting information about possibly the produced molecules by these organisms. Of course, some people started to sequence many different environments, and this is the case of this one paper from 2016. And actually, what was amazing there is that they went to this deep aquifer system, and what they could do, they achieved to reconstruct more than 2,500 near complete genomes. So that was a lot. But then when they performed phylogeny, they also realized that they were covering lineages that were never sequenced before. So they were also even proposing up to 47 newly discovered phyla. So I mean that was huge in terms of the diversity being covered here. Again, this is a tree, I won't spend so much time here, but it's a tree that actually shows where they could contribute new genomes, and there are novel lineages being represented. And in blue, actually, there are lineages that are lacking a genomically characterized organism. So those were not found in this study, but all the black ones, what that means that it covers most of the diversity, they actually did find representative in this deep aquifer system. And not only that, I mean, they have access to genomes, but then, of course, what to make sense of it, of course, it represents, it helps to have a better representation of the diversity of the organisms in this aquifer system. But not only that, I mean, from the genomes, you can also predict the gene function and try to understand what are the processes at hand in this environments. And what they did is that they actually predicted, they focused on different elemental cycles here, and they found actually enzymes contributed to, for instance, the sulfur cycle here, nitrogen cycle, carbon cycle, and they show how the different organisms interconnect and contribute to different parts of these elemental cycles. So, with this one study, actually, from deep aquifers, of course, they had access to genomic information for uncultured lineages. They were covering a lot of the diversity and underexplored environment of this subterranean environment. And also they discovered metabolic diversity, and it helps actually to understand the global biogeochemical cycles and the contribution of the different organisms to that when you have access to the genomes. Another example, and which is also another type of study, we don't all have to go to the field and collect new samples to make discoveries. So this is why I like this study. Actually, this team, they reused actually the metagenomic data publicly available, and they designed a pipeline to reassemble, to bin the genomes from this publicly available data. And it was actually striking that with the technological progresses being made, they could actually extract more information than was done first when these public datasets were published. So you can also do this kind of meta-analysis and still make very interesting discoveries. So in this particular case, actually, they say that they could report the reconstruction of nearly 8,000 bacterial and archaeal genomes from more than 1,500 public datasets. And also they say again, so you know the drill now, they actually wanted to show how much they would cover in terms of diversity, but first here it was more about assessing the quality of the genomes recovered from these studies. And what they are showing is that so here is shown the completeness of the genomes versus the contamination. Of course, you want to, when you bin genomes, you might take contigs from different organisms, so there is a risk for you to have contaminations. What they could show here is that they had actually a high portions of the genomes recovered that would be very highly complete, actually almost near completion, while being still a little bit only contaminated, which would be actually a good measure to think that they would be high quality genomes and that you can somehow trust these genomes. And again, the classical figure here would be to show on the tree how this study contributed to enhance our view of the biodiversity. And actually here they show that they would cover like most of the lineages known, that is in green in this study, but they would also discover new lineages, and those are the candidate phyla that they proposed in red. So this is for the bacterial side of life, but they also showed the same for the archaeal side of life, where they also proposed new lineages. Okay, so as I said, from the reanalysis of publicly available metagenomes, you can make some interesting discoveries anyway, and discover new lineages in bacteria and archaea. So now, okay, we've seen that these genomes, we can place them on trees and try to understand the biodiversity. But what does it really tell us about life history? So first of all, you've seen that there were all these different studies. It was almost at the same time, actually. It was actually a lot. It was a lot of studies that would come out and claim that there would be new lineages and so on. And somehow it also seems like a gold rush where people would claim that they found a new lineage, etc. So of course you also at some point want to make sense of all of it and combine all the information. So there are actually some consortia that discussed how to name properly the lineages, how to classify the different lineages that would be maybe more in relationships to a tree of life, etc. So that's another matter. But anyway, then in 2016, there were this paper that came out. I'm showing you as a reminder the 1993, where are shown all the different phyla that were known then. So that's the Worth and colleagues paper. And this is the view that this article showed, to the point that it was called a new view of the tree of life. So it came out in 2016, and here are shown actually all the different phyla that was proposed then. So you see how much of the diversity was now put in this one tree of life compared to what was known in 1990. So in such a tiny amount of time in the end. And actually what was really striking is that this part is actually, this entire part here is called the candidate phyla radiation. It was actually not known so far. So it's been like an entire part of the tree of life that had been somehow ignored, and where its biodiversity was not really acknowledged. So yes, so tree of life, you have the bacteria here, and with the dots, the red dots are shown the culture, no, it's not that, sorry. Okay, forget it. Anyway, so you have the bacteria in blue here in this part, and archaea here in red, and eukaryotes here. So that's the tree of life. Okay, so of course there is this one candidate phyla radiation that was totally overlooked so far, but actually not so much overlooked, because actually people realized afterwards that some isolated lineages would have been observed and would be part of this candidate phyla radiation. That was the case of these ultra small bacterial cells. And one of the reasons that this part of the tree of life has been overlooked is for a very simple reason that when you go to the field, you would do some filtering. For instance, if you filter water, you want to collect some biomass, well, you will use different filters so that you will fractionate your samples. For instance, for the viral part, the bacterial part, the eukaryotic part, and so on. Well, some of the bacteria were maybe tinier than we expected, and they were overlooked for these simple reasons that they would pass through the filters. So, yeah, of course, sampler bias, it exists, and in this case, it was striking, actually, to see that it could be due to that. So all these candidate phyla radiation, most of them anyway, they are tiny cells, probably, so they are not cultures, so we don't know exactly, but from the genome sequences also they have tinier genomes, and this is why probably they were overlooked. Yeah, so those are some cryo-electron tomography. Oops, sorry about that. Okay, so this tree of life. Well, oh, I forgot to mention, we are humans. We are here. Okay. So this is where we are, and actually it is even more striking when you look into this rectangular view of the tree of life, where each major lineage represents the same amount of evolutionary distance. So to say it's somehow representative of the diversity. You see that eukaryotes here are just a tiny fraction of the diversity, and we are just one branch within eukaryotes. So I like this because it's also put us into place in the tree of life. Okay, most of life is, well, it is bacteria, it is archaea, and then eukaryotes. Okay, so of course, microbes and humans, it's been a very long story. They have been studied, the microbes, I would say mostly bacteria, because they are responsible, of course, of many pests and diseases. So we have also a very anthropocentric knowledge somehow of many of them. They've been observed as early as the 17th century, and as you may know, there are as many microbial cells than the human cells in our own body. Some have even co-evolved with humans, and well, we derive from microbial ancestors, and this is where I want you to, this is where I will continue on this topic of our own ancestry. So what I find really interesting and exciting about microbial forms is that bacteria and archaea actually they colonized all of these different environments, very different environments, and this is thanks to their very highly diverse metabolisms. They are able to live acidic environments, in alkaline or high salt concentration environments. They are found in peatland soils. They are found even in symbiosis, for instance, with sponges, in water, soils, agricultural soils or not, natural or even anthropocized environments. And even you find them on the human skin, for instance. So I think it's very fascinating to observe that and to understand how they came about all this diversity that make them adapted to such different environments. But also bacteria, archaea, they contribute a lot to biogeochemical cycles, and this is the case, for instance, of cyanobacteria. And as you may know, cyanobacteria, they evolved oxygenic photosynthesis, which was actually crucial for the great oxidation event. So in this graph, you see here the pressure, the atmospheric pressure of dioxygen. So this is a model. There are more recent ones, I think, now. But anyway, and in blue you have the this is the error rate of the model. But what you see is that when cyanobacteria evolves oxygen photosynthesis, at some point dioxygen was produced and it accumulated, and this is held actually to this led to this great oxidation level event, where the level of dioxygen increased tremendously. And we can link this with of course, the expansion of aerobic respiration, meaning breathing dioxygen, because we are not the only ones to breathe dioxygen. Many bacteria actually do. And also this later increase in dioxygen levels is somehow linked to animal diversification. So bacteria are responsible for the air that we breathe. But not only, actually. There were many contributions to what we are now from bacteria and archaea. And this went through the process what is called endosymbiosis. And I will call them the ultimate co-options, where actually you have a cell. Here, for instance, this is the case for the origin of mitochondria. You have a host cell that actually engulfed probably ancient protobacterium, or now called Pseudomonadota, anyway. And this actually engulfed cell will give rise to what is now mitochondria in our cells. And these cells would have been somehow domesticated for energy production. Actually, we produce ATP thanks to this mitochondria. And this actually happened several times along evolution. And for instance, this also came about before the diversification of the green lineage, where the plastids arose from the endosymbiosis by an ancestral eukaryote of a cyanobacterium. So cyanobacteria were engulfed in another endosymbiotic event, and this gave rise actually to plastids, which are responsible for photosynthesis in plants. So you see that those are very important contributions of bacteria to eukaryotic lineages diversification. Also, I just wanted to point this paper by Lynn Sagan-Ven, and after she was Lynn Margulis, on the origin of mitosing cells, where she was already somehow, even though she didn't have access to all of this fancy genetic data and genomes available to build phylogenies, she proposed theory of endosymbiotic theories about the origins of eukaryotic cells, for the origins of the mitochondria, but also of the plastids. So for instance, what do we know about the exact origin of the plastids? It goes through the sampling of new organisms and also building phylogenies and trying to position cyanobacteria versus plastids. And this is what our colleagues from Orsay did, actually, in this one paper from 2016. They actually isolated a cyanobacterium from freshwater environment, and they actually managed to culture it in the lab. And what they could see is that this Gloeomargarita lithophora, I think it's beautiful as well. What they showed is that it would position, this is a tree of cyanobacteria, and this one bacterium, cyanobacterium, would actually branch next to photosynthetic lineages, the green lineages in eukaryotes. So it means that actually those are the closest extant lineage to the ancestor of the plastid. Another actually study from 2015 started to obtain genomes from obscure archaeal lineages that would be called at that time DSAG or MBGB for Marine Benthic Group B. So they were known, their existence was known from DNA evidence. 16S ribosomal RNA would have been sequenced for them. So people would know already that they would exist. And actually, this one study that was previously published was actually reporting the existence of these organisms in sediments at the Arctic mid-ocean spreading ridge. So that would be sediments that would be influenced by this the hydrothermal activity of these vents here at a position that is called Loki's Castle. So that would be actually here. So Norway is here. So that would be where they would have sampled this. So this study from 2015 started to obtain genomes for these obscure groups. And actually, to their big surprise, I guess, what they realized once they obtained these genomes, when they would reconstruct a tree, is that these newly obtained genomes that they called Lokiarchaea, they would actually branch next to eukaryotes. So that would be archaea that would be closely related to eukaryotes. And okay, because we may not want to trust only a phylogenetic tree because, as I said, it is difficult sometimes to build them. You see that this is the statistical support. I mean, one is it's a probability, so it's the highest support, but 80, it's over 100, so it's not the greatest support. So how do we trust it anyway? Well, we spoke about phylogenetic systematics before. You may want to go back and find, if you find from these genomes shared derived characters, shared derived traits. So this is what they did. On top of these phylogenies, they investigated the genomic content of these newly described genomes of archaea, what they called Lokiarchaeota. And they actually hunted for particular proteins, protein-coding genes in the genomes, the one that was actually thought to be specific to eukaryotes, meaning that these eukaryotic specific proteins, or ESPs, so far they would only be found randomly, seldomly, in other lineages from that eukaryotes. They would be specific to eukaryotes. So this is what is represented. You have a dot for each of these ESP. And you see that most of the archaeal lineages, they would hardly have one or a few of these ESPs. But the newly discovered archaeal lineages, well, the ones for, well, the newly obtained genomes for this archaeal lineage, because I said they were known before, but we didn't have access to the genomes, they would show the presence of many of these ESPs. And it would be the first time that as many ESPs would be assembled together in an archaeal genome. And this is another evidence that this lineage of archaea is actually closely related to eukaryotes. And this is where actually, that was the title of this one paper from 2015. it's called complex archaea that bridged the gap between prokaryotes and eukaryotes. So it was a huge discovery. And following up on this, actually, people continued to refine because there would be already theories about the origins of eukaryotes that would be called eukaryogenesis. Well, the new thing here is that we would have a better sense of the identity of the ancestral lineage that gave rise to eukaryotes. So basically, remember we talked about in the symbiosis, so that would be about the symbiotic origins of eukaryotes, while you will have this eukaryote Asgard common ancestor that would merge with this ancestor of the mitochondria, as I explained later on. That would lead to this cellular merger and into the first eukaryotic cells. As also, environmental studies and researches went on, of course, people started to hunt for more and more of these closely related archaeal lineages with respect to eukaryotes. And the tree of these Asgard archaea, actually, they were called like this because Loki's castle, Loki Archaeota, Loki is actually from the Norse mythology, and they started to find more diversity of this Asgard archaea with other lineages that would even be more tightly connected to eukaryotes. So here is actually it represents the diversity that is known. It's from 2023, so it's not so old, but I mean, people are still working on that. But here, in this one paper, actually, and you see that there was this phylogenetic study that showed that actually that would be hodarchial, that would be the most closely related to eukarya. And to give you an idea, well, Loki Archaeota, they are here, right? So people found even more closely related lineages to the Loki Archaeota then. And if we go back to the ESPs now, well, the list has been enriched. And again, so that would be the proteins that would be more likely to be specific to eukaryotes so far, these ESPs, with these dots. And you see here the first line is Loki archaeal. And actually, so it's filled when it's present. You see that the hodarchial, which are the ones that are more closely related in the tree, they are also the one that fills more of these dots, meaning that they have more of these ESPs. So even further evidence that they are really more closely related to eukaryotes. Okay, and so what is also fascinating, and I think it's also really rewarding hard effort. So I won't show you, I'm just super excited about this one preprint that just came out. But actually, it was really nice to see that sometimes years of effort can be rewarded. So this group, this Japanese group, actually, by coincidence, they were trying to enrich these obscure archaeal groups called DSAG. Anyway, so they were the first ones to release, to obtain in culture, in enriched culture, the first Loki Archaeota. So this is not this paper. This one paper was from 2018 already. But what I'm showing you here today, in this study, actually, they said that it was years and years and effort, and then people realized that they were super interesting, and they were the ones that had made the efforts already before, and they had the first culture of this Loki Archaeota, because of course many people are now interested in getting them in culture as well, to study them more closely. And they released actually, like less than a month ago, this preprint where they actually say that after 3 years of effort, they obtained a culture of hodarchial, meaning that this is this time the most closely related lineage to eukaryotes. So yeah, I couldn't manage to not include these beautiful microscopic pictures of these hodarchials. Actually, they made the isolation of 2 of these strains. I say isolation, but actually they live in syntrophy, meaning that they live in tight connection with other organisms. So one of the strains has been enriched with another organism, which is a methanogenic archaea. So it is a co-culture. And the second strain of Audarchaea has been enriched in a tree culture, actually, with 2 other lineages, one being also methanogen, or maybe even the 2 ones, anyway. So they live in tight connections with that. And so you can see them here, actually, and I didn't put the scale. I don't have it in mind. They are not super large cells. I'm sorry, I don't have the scale here. But anyway, so also what was remarkable compared to the 2 first cultures, because there were 2 first cultures of Aulochiarchaeota before this one, is that they seems to demonstrate, they say, a behavior focused more on cell construction rather than division. They are very slow growers, and they, when they divide, you see they are more like cocoid shaped. And actually, when they are not dividing, and they have these protrusions here that form, that are actually, they consist in a very high volume in the end of the cell, this protrusion. And what they also observed is that this protrusion here are involved in the connection with the other syntrophic organisms in the culture. And it seems even that there might be some pili that are involved in the connection between the protrusion and the methanogenic archaea present in the culture. So they make some assumptions from the genomes on the metabolism and what would be the possible exchanges between the different partners in this culture. So I think it's very interesting and very promising. And why is it so important actually to have these organisms in culture is exactly for that, to try to understand their physiology and their behavior and how they interact with other organisms, for instance, in this co-culture or triculture. So basically, really, I mean, getting all these genomes is very important, of course, but what would be also very interesting is to try to have them in culture and try to understand how we could actually really understand in depth the process of eukaryogenesis. So if you're interested, you can read this article by our colleague from Orsay about the different hypotheses on the symbiotic origins of eukaryotes. And you see that we have the biggest contributions from archaea, from alpha proteobacteria with the mitochondria, but there might be also other partners involved, and the fact that these cultures are showing actually tight connection and syntrophic behavior for these lineages that are more closely related to eukaryotes points probably to some syntrophic hypothesis for the origins of eukaryotes. And I think this is exciting times ahead, and people will try to manage to understand our past ancestors based on these contemporary organisms. Okay, so I think I have a little bit of time, maybe just to conclude. So also, all of this, it also tells us a story about the place of some organisms compared to others. So I spoke about this obscure archaeal lineage that was somehow, people got interested into them, but then people realized that, okay, well, they are the cousins to our ancestors, so wow, this is very super interesting. But yeah, people picked an interest anyway in this, so this tells us a lot also about curiosity-driven science, and I really like it about that. And also it tells us about the place of archaea a long time. So when you hear about archaea, you might think about extremophiles and life in extreme environments. And of course, actually, you can see that archaea have capabilities to live in extreme environment on different sides of the thermometer, of the pH scale, etc. Some cannot even tolerate dioxygen, like the methanogens, for instance. Yes, so some of them are extremophiles, but not all of them, actually. And people actually realized, so this is a tree of archaea where you have a color gradient showing the optimal growth temperature. And this is from a 2011 study where people actually reconstructed the optimal growth temperature of ancestors. And the idea is to try to figure out whether the ancestor of archaea was, for instance, hyperthermophile, thermophile, mesophile, meaning living at high or lower temperatures. So people, of course, started to realize that they were not all thermophilic organisms. You have also, for instance, the form Archaeota, but now even further, that are lineages that are being mesophilic lineages. And they can also, for instance, inhabit here the sponge, Axinella mexicana. So this is the case of the Formarchaeota Synachium symbiosum. So they can also establish connection with eukaryotic organism. And also, along the time, people realized that they're also part of the microbiota in humans, for instance, not only humans, but also other animals, etc. And actually, this archaeome somehow has been neglected, and some studies from 10 years ago, they showed that it was again somehow a sampling bias, so to say, because the molecular probes that were used to fish for the presence of different organisms, they would actually not catch all the diversity of archaea. So we were somehow overlooking also the presence of some organisms in the microbiota. So just to tell you that some of us have actually also archaea in our microbiome. And something that is also very important, I spoke about the place of bacteria in biogeochemical cycles. It's also the case for archaea, which are involved in many different parts of the biogeochemical cycle, the carbon cycle. So in orange, you have the parts that are only performed by archaea, the nitrogen cycle. So in red, you have parts that are performed both by archaea and bacteria, and the sulfur cycle, for instance. So yes, I mean, archaea matter. And with this increasing view of the tree of life, we also saw that they are more diverse than we thought. And of course, we spoke about the place of archaea in the contribution to eukaryogenesis. Okay, so I think I already made my point here. So to conclude, I wanted to somehow make a recap of some of the pending questions that we have still about the tree of life, and not only about the tree of life. It's actually also about what are doing all these organisms on Earth. I mean, obtaining a tree of life is something, trying to make sense actually of the respective roles of the different organisms is another thing. And actually, I think we made tremendous progress towards understanding the origin of biodiversity, but also this gives substrate to study what I called earlier molecular evolution, try to understand the arisal of new biological functions. Our own origins already make a strong point, I guess, about it. So yeah, so among the pending questions are, for instance, predict the function of genes from genomes. That's a huge task. I'm just summing up a lot like this, but it's an entire open question here. I mean, we have some clues but I mean, this is a huge task actually. Understanding the trees of eukaryotes, of animals even, it's not solved. The origins of multicellularity, the origins of bacteria and archaea. We spoke about eukaryotes, what about archaea and bacteria? Also, how to root all these trees, because we want to also understand, orient them in time and understand what are the most ancient part of the tree. So there are all these questions to address, and of course so many more. And to do that, of course, we also, it's like a circle, we will also want to understand, but at the same time we need to build the tree of life to be able to make sense of all this data. So how do we do that? But I think that one way is actually, of course, to continue combining computational and experimental approaches. Computational approaches, I hope I convinced you that, yeah, they are actually required to make sense of all this data. But of course, experimental approaches are really requested, and to actually dig into the actual ecophysiology of all these organisms and understand what they are really doing. And yeah, I think this is just by combining this that we can manage to make sense of this tree of life. And of course, needless to say, but most of this discovery, all of these discoveries are due to curiosity-driven and interdisciplinary basic science. Yeah, and I think, yeah, this this is how I'd end, I guess, my talk. Thank you.